The next presentation will be by Panagiotis Theodorou. I should say Panas because that's easier for me. And it's on Bombus Terrestris colony performance in the urban ecosystem. Panas, can you start? Yeah, thank you.
All right. I hope everything is fine with the presentation and with the sound.
It's good, yeah.
Perfect. So, good morning. My name is Panagiotis Theodorou. I work at the Martin Luther University in Halle, in Germany. And today I'm going to present to you a study which is investigating the main environmental correlates of Bombus terrestris colony performance within the urban ecosystem. As we all know, animal pollinators are important components of biodiversity. However, in the past decades, several reports have highlighted and documented pollinator declines, both locally and globally, declines that could have major ecological and economic consequences. Here you can see listed the 4 major threats to pollinator biodiversity. In the top of the list is land use intensification and anthropogenic habitat alteration and degradation, resulting from the convention of semi-natural to human-dominated habitats, including agricultural and urban. Urbanization is an important driver of land use change. However, compared to agricultural systems, pollinators in urban areas remain relatively unstudied. This is not surprising, given that approximately 38% of the Earth's ice-free landmass is used for agricultural purposes, and only 8% is urbanized, suburbanized. Nonetheless, the urban global cover and the urban global human population around the world is increasing. As you can see from this figure here, which in the y-axis we have the percent increase, both of the global urban human population and urban land cover. With dark green, we have the urban global population increase, and with green, we have the urban land cover increase. So from this figure here, you see that from 2000 to 2030, the percent increase in global urban land cover will be over 200%, whereas the global urban human population will grow over 70%. As cities are expanding worldwide, urbanization has been identified as one of the major threats to global biodiversity, as well as a driver of evolutionary change. Impacts of urbanization combine both the rapid destruction of natural habitats and their slow degradation, the increase in impervious surfaces, fragmentation of natural vegetation, invasion or cultivation of non-native species, along with long-term environmental effects such as the heat island effects and soil and air pollution. All these are working in concert, resulting in the creation of novel ecosystems that exhibit novel species composition. The creation of such novel habitats could be challenging for both the persistence and survival of many species, including essential ecosystem service providers like insect pollinators, whilst also imposing altered selective regimes. Nonetheless, in moderately disturbed urban habitats, some species, including several pollinator taxa, are doing particularly well. And that's because of habitat heterogeneity within moderately disturbed urban areas, the high availability of food and nesting resources, and continuity of floral food resources within the moderately disturbed urban areas. Overall, indeed, the effects of urbanization are complex and depend on the type of change, whether it's moderate or intensive, the taxonomic group studied, and the scale of investigation. Urban areas overall encompass tremendous heterogeneity across both spatial and temporal scales, and are therefore unique macrocosms for studying a pseudo-fundamental and applied biological questions. The number of studies investigating big community patterns in cities is increasing, as you can see in this figure here, which in the y-axis we have the records from the Web of Science, and in the x-axis we have the publication here. As you can see, this relationship is exponential. So the number of studies investigating bees in cities is increasing. And those studies, those community level studies, give reason to view urban environments as refuge for species affected by agricultural intensification, revealing the importance and potential of cities for bee conservation. However, we lack studies investigating the effects of land use, of urban land use on fitness, the ultimate driver of ecological success and evolutionary change. Among the wild pollinators, bumblebees have become a model system for studies on behavior, ecology, and evolution, partly because of their importance, ecological and economic. The majority of bumblebee species are primitively eusocial and generalist pollinators and can therefore facilitate the reproduction of a large number of wild plants and crops in diverse terrestrial ecosystems, including urban. However, they are currently facing a number of threats that include changes in the floral resource availability associated with land use change and climate change. Today, I will present to you an experimental study we did investigating Bombus terrestris foraging behavior, parasite prevalence, and colony fitness within the heterogeneous urban ecosystem. Before I continue with presenting you our research, I would like to thank my colleague, Dr. Christoph Kurze, the two master students involved in the study, Olga Kuhn and Lucy Baltz, as well as Christopher Wilde for all their hard work in this project that I will present to you today. So let's start with the first part of my talk, which is in regards to the main environmental correlates of Bombus terrestris parasitism levels within the urban ecosystem. All parts of my talk were addressed by studying Bombus terrestris experimental colonies in 10 sites within the city of Halle in Germany. All sites were pre-selected, spanning from the edges of the city and into the city's core to represent the environmental heterogeneity within the city of Halle as much as possible, and to ensure spatial independence, sites were selected to be at least 1 kilometer away from each other. So what we did, in the beginning of May, we bought 20 commercial Bombus terrestris colonies. Upon arrival, those colonies were standardized to have 20 workers. Each of those workers was then individually labeled and placed back to their colony. And all the colonies were placed in individual styrofoam boxes, as you can see in the bottom of this slide, with ventilation holes. And we did that in order to protect them from rain and animal damage when we placed them in our 10 experimental locations. So what we did, we took 2 colonies with their styrofoam boxes, and we placed them in partially shaded locations in each of those pre-selected 10 sites, and the monitoring of colonies started, and we ended the monitoring in the 7th week after placement in the field. In each of these locations, we documented a number of local and landscape scale variables that could potentially affect Bombus terrestris foraging behavior, parasite prevalence, and colony performance and fitness. So in each location, we documented the presence of honeybee hives, and every 2nd week, we use a scanning method to assess local flowering plant richness and abundance. We furthermore use quantum GIS and land cover maps to characterize the landscape surrounding our experimental colonies. We estimated a number of landscape scale variables, including road density, the proportion of green cover surrounding our experimental locations, edge density, which is the number of ecotones, which is defined as the total length of edges surrounding our experimental site. And we also calculated the distance from the city center as a variable that encapsulates the intensity of urbanization. And all these landscape scale variables, together with the local scale variables, were included as potential predictors in all our downstream analysis. In addition to the local scale and landscape scale data, we also used thermaloggers inside each styrofoam box and recorded temperature between 7 to 5 p.m. Colonies were also weighted weekly, and we used that as an indication of colony growth. And for this part of my talk, 3 returning foragers per colony and week were sampled for parasite screening. We screened a total of 583 returning foragers for Crithidia bombi, Nosema bombi, and Apicystis bombi using the PCR methods. All these three parasites are very common protozoan endoparasites of bumblebees, which are potentially threatening bumblebee colonies and might contribute to their decline. Here, I show you a schematic revealing the differences in the local and the landscape scale variables across these 10 locations. In the bottom of this figure, you see the locations from site 1 to site 10, with site 1 being the closest downtown and site 10 being at the edge of the city at 6 kilometers distance from the city center. And above this schematic of the city, you see the range of values of floral abundance and richness, of semi-natural cover temperature recorded within the styrofoam box, and road density. And as you can see, most of these variables here varied quite a bit. The road density differs fourfold from the edges of the city and into the city core. Temperature 1.3-fold increased from the edges of the city and into the city's core. And flowering plant richness, flowering plant abundance, and semi-natural cover also varied from the locations downtown that we placed those colonies to the locations that they were at the edge of the city. So we had a lot of variability in our local and landscape scale metrics. So let's go back to the parasite prevalence. And here, which were the parasite prevalent levels of our experimental colonies. As you can see from this figure, we did not detect any individual infected with Nosema bombi. In the y-axis here, we have Nosema bombi parasite prevalence, and in the x-axis, we have the weeks of sampling. And as you can see from this figure, no individuals were found to harbor Nosema bombi. In regards to Crithidea bombi prevalence, as you can see from this figure here, in the y-axis we have Crithidea bombi prevalence in percentage of infected foragers, and in the x-axis we have the week. The colonies as they were placed in each of these experimental locations, in each of these locations in week 0, there were no infected individuals. And as the weeks pass, as you can see from this figure, we have almost 100% infections in certain locations, and the average at about the 5th, 6th week is 75%. In each color and shape here corresponds to one of our 10 locations. Similar results we obtained for Parasitism bombi. Again, 0 infections as we place them in each of these 10 locations, and then the infectious rate rise up to the 5th and 6th week, which we see also about 75% of infected individuals across those 10 locations. As both of Crithidea bombi and Parasitism bombi, they were highly correlated. Today, I'm going to present to you only the results for Crithidea bombi. So using a generalized linear mixed effects model and a model selection approach based on NAIC, from all the potential local and landscape scale environmental predictors, we found that colony growth and distance from the city center were the best predictors of Crithidea bombi parasite prevalence. Crithidea bombi prevalence increased with an increase in colony growth, as you can see in the first figure here. In the y-axis, we have Crithidea bombi prevalence, and in the x-axis, we have colony growth. This probably reflects the higher probability to bring new infections to the colony and the high probability of an uninfected bee to get in direct contact with an infected bee as the colony size increases. And in the second figure, you see the relationship between Crithidea bombi prevalence in the y-axis and in the x-axis distance from the city center. So here we found that Crithidea bombi prevalence increased in colonies that they were more distant to the city center. Across host parasite systems, parasite prevalence is influenced by host population size and vice versa. Bumblebee density and greater rates of pathogen transmission might be higher in the edges of the city compared to the city center, which could result in increased Crithidea bombi prevalence of our experimental colonies. Next, using the same experimental colonies, we investigated the main environmental correlates of Bombus terrestris foraging behavior within the urban ecosystem. To do so, above each entrance in each of the colony, we used GoPro cameras and we recorded bumblebee traffic for 6 hours per week and colony. Today, I'm going to present to you our estimates of foraging trip duration. For this, we analyzed over 200 hours of video recordings of the first 2 weeks. In those videos, we observed and recorded the time a marked bee left for foraging and the time it returned, and we use it as a measure of trip duration. We only analyze here the first 2 weeks because after those 2 weeks, we could not detect anymore any initially labeled worker. So again, using a generalized linear mixed model and a model selection approach to investigate main environmental correlates of foraging trip duration from all those potential environmental predictors, edge density and the presence of honeybee hives were the best predictors of foraging trip duration. Foraging trips were shorter with an increase in edge density. As you can see in this first figure, in the y-axis, we have the foraging trip duration in seconds, and in the x-axis, we have edge density, in this case, in 750 meters radius. And as you can see, there is a negative relationship. With increasing edge density, there is a decrease in foraging trip duration. And this suggests that ecotones, or edges, might be very useful foraging habitats and foraging routes for bumblebees, decreasing consequently their trip duration. Foraging trips were longer, as you can see in the second figure, with the trip duration again in the y-axis, and in the x-axis we have the presence of honeybees. So foraging trips were longer at sites with honeybee hives, suggesting that honeybees might compete and drive bumblebees away from nearby floral resource-rich patches, resulting in an increase in bumblebee foraging trip duration. The last part of my talk, using the same experimental colonies, we now investigated the main correlates of Bombus terrestris colony growth and fitness within the heterogeneous urban ecosystem. If you remember, in each colony, we used a thermalauger to record temperature, and we weekly measured colony weight, and we used it as a metric of colony growth. In addition, for this part of the research we did, we also measured the number of giants each colony produced, and we used that as a proxy for a colony's reproductive success. Here you can see the differences in colony weight between each of our locations. In the y-axis, we have the weight in kilograms, and in the x-axis, we have the week of sampling. And as you can see in the beginning of our experiment, since we standardized colonies down to 20 workers, all of them had the same initial conditions and the initial weight conditions. And as you can see with the weeks passing by, we see a lot of variation in the colony weight, reaching its peak at about 5 to 6 weeks. And you see the 7th week in which we see a drop in colony weight almost from all colonies. So again, using a mixed model approach and a model selection approach for all our potential predictors of colony weight, average temperature across the sampling period was the best predictor. What we found here, as you can see from this figure, in the y-axis we have the weight in kilograms, and in the x-axis we have the temperature in degrees of Celsius. Bombus terrestris colony weight decreased with increasing temperature. This decrease in colony could be due to workers switching from foraging and brood care to nest fanning under high temperatures. Here I present the average temperature across the entire sampling period, but we run the analysis also with just using the maximum recorded temperature in each of the locations, and still we find the same outcome. Using again a model selection approach to investigate what are the main correlates of giant production, what we found that colony weight was the best predictor of giant production across our 10 locations. As you can see from this figure, number of giants is in the y-axis and colony weight is in the x-axis. So this positive relationship has been already documented in a large number of studies, revealing that colonies of larger size invest more in new queens. And more importantly, after we run a structural equation modeling, our results suggest that there are indirect negative effects of temperature on reproductive output via its negative effects on colony weight. To sum up, Bombus terrestris parasite prevalence increased significantly with colony growth and distance from the city center, probably driven by an increase of potential hosts within the colony and in the outskirts of the city. Bombus terrestris foraging trip duration decreased with decreasing edge density, but increases with the presence of honeybee hives. Ecotones might be very useful foraging routes that provide connectivity among foraging habitats for bumblebees, and honeybees and bumblebees might compete for resources. In regards to Bombus terrestris colony growth and fitness, we found that increasing temperatures could have a negative impact in slowing colony weight and indirectly in reducing colony reproduction. As an outlook, our results point to the importance of ecotones for bumblebees, and such habitats should be considered in city planning. Furthermore, due to the potential competition effects of honeybee hives and bumblebees and the increase in urban beekeeping in all major cities, as you can see here, as an example, in Berlin, we have more than 10,000 honeybee hives. What I would suggest is that stakeholders should take into account the impacts that apiaries could have on the wild bee fauna, including bumblebees within cities. And at last, there is an urgent need to control the heat island effect within cities, and this could be done with planting trees and other vegetation to lower surface temperatures within cities. And with that, I would like to thank the entire general zoology group at the Martin Luther University in Halle, and all of you for your attention. Thank you.
Okay, thank you for this, this very nice and very clear presentation. I saw on the chat that there was some discussion on whether or not it's good to introduce Bombus terrestris colonies in natural environments. And people were also discussing whether or not these colonies which you buy can already have parasites in them.
Yes, it's generally not ethical to introduce commercial colonies in the wild, especially that could lead to escapes of queens or males and so on. So what we did to avoid such problems, colonies were monitored, and that's why we terminated the experiment at the 7th week and did not continue until the death of the colony in the field. So colonies were monitored and they were removed from the field with the first signs of giant production. So we tried to avoid such a potentially terrible byproduct of such an experiment. All colonies that we got from this commercial provider, they were free of Crithidia bombi, Nosema bombi, and Apicystis bombi. And that was done by us. When we received them, we screened them, and then we put them out in the field. If we have detected an infected colony, that will have biased, obviously, our experiment. And obviously, we will have excluded it from the placement in the field. So to answer that question, no colonies were infected with these 3 parasites.
Okay, thank you.
In the beginning of the experiment.
There's another question by Julia Pico, and that's about the results you had on the impact of honeybees, which is of course very interesting. And the question is, could the presence of honeybees hives also have an indirect positive effect if beekeepers provide additional flower resources?
That is not usually the case with beekeepers. It's not that they also manage the land to provide resources for their bees. Usually the beekeepers, the urban beekeepers, are doing so as a hobby to produce their own honey. And given the high amount of floral resources already in cities, such a management practice from beekeepers is not the case. So if our aim is to conserve wild bee biodiversity in cities, and the ecosystem of pollination, obviously, they provide, then I will suggest that we need to manage the number of honeybee hives and of apiaries in general in our cities to avoid such competition effects. Bees are facing a lot of problems in the rural ecosystem, in the agricultural ecosystems, and from what we see from community level studies, the cities could provide potential refuge for wild bees affected by agricultural intensification. And at the same time, they could act as a reservoir to populations that they exist in the rural ecosystem. So if our view is to manage and conserve wild bee fauna in cities, then surely we need to find a way to manage urban beekeeping.
Okay, that's clear. One last question. Did you look at the new queens, if there is a strong correlation in the number of parasites they have and the number of parasites in the colonies itself?
Unfortunately, we didn't have a look at that. We still have the queens in ethanol in the lab. So we will certainly try to do that indeed. I will say that since they are reared in the same environment with workers that we sampled from, I would say that they will reflect the levels of parasitism in the colony that they were found in. So I wouldn't expect differences between the last points of infection levels of the colony and the infection levels of the queens from those specific colonies.
Okay, thank you. That's clear.